Hello students, so let's talk about the urinary system. So the urinary system is pretty important as a fundamental piece of our anatomy and also our physiology. So the kidneys are very important for us. So they help us removing salts and metabolic waste through the urine, and also they help us regulating blood volume and blood pressure, okay? And they also helps us regulate and maintain a normal water concentration versus electrolyte concentration. This is known as osmolarity, okay? So thinking about the amount of fluid versus the amount of concentrates or solutes that are in our blood, such as sodium, potassium, so on, so on. And also ensures long-term pH levels in our body. We have talked many times about the concentration and good pH in our body because this is important for optimal functioning of our bodies. The kidneys also produce a very important hormone called erythropoietin, also known as EPO, and this one controls the production of red blood cells, okay? And there's also the production of a hormone called renin. Renin is a very important piece in our blood pressure and volume balance because it activates the renin-angiotensin aldosterone pathway, okay? With the end result of controlling our blood pressure. So in addition to the kidneys, we have other structures in body systems that helps us regulate those metabolic wastes. We are very familiar with sweat. We are very familiar with the carbon dioxide being expired through our gas exchange, but also there's water vapor going on through our respiration. And also in the digestive system, the large intestine contains some digestive food, water, and salts. Okay? So components of the urinary system are very straightforward. We are going to concentrate highly in the kidneys, but also we cannot forget that we have 2 kidneys, 2 ureters, 1 urinary bladder, and 1 urethra. This is regardless of biological gender, okay? All right, so the location of the kidneys, they are located posteriorly level T12 to L3. They are retroperitoneal because they're behind the peritoneal cavity, okay? The right one is lower than the left one because the liver is taking a lot of space. And basically the kidneys are the stars of the show. The other pieces, meaning ureters, meaning bladder, or meaning the urethra, they're more or either a conduit, meaning passageways or storage of urine and waste. So what are the connective tissues covering the kidneys? So all kidneys, meaning 2 of them, have a renal fascia. This is an outer dense layer that anchors the kidneys in the adrenal glands. Pretty important because we cannot forget the kidneys are accompanied by the adrenal glands on top of them. And these layers of fascia attach the kidneys to the abdominal wall and surrounding structures, okay? And there's also a perirenal fat capsule. This one works as cushion and protection for the kidneys, okay? And then we have talked about the outer layer, the renal capsule. This one is a transparent fibrous tissue that protects the kidneys from infection spreading from other locations, okay? Some quick facts about the anatomy of the kidneys. So kidneys are approximately 11 centimeters by 6 centimeters by 3 centimeters, and they weigh only 5 ounces. They're kind of like light for the big impact that they do to our bodies. So the medial surface, meaning the area that is inwards, is concave. And we're talking about this area here, right? And then the lateral surface, meaning this area here, as you guys can see, is it's convex, all right? Convex. As we talked about the lungs and the spleen, they have a renal hilum. This is the entrance and exit point for blood supply, lymphatic vessels, and nerves into the kidney, okay? And on top of our kidneys is where the adrenal glands sit. So given the importance of our kidneys filter in our blood, during resting state, we receive from 20 to 25% of our cardiac output to the kidneys. So the right kidney receives the blood from the renal artery, right? And the left one, of course, gets it from the renal left artery. So, as we were talking, because of the position of the right kidney, the right renal artery is going to be longer than the left because of the position, right? The other thing is that these renal arteries are branches of the abdominal aorta, and you can trace it back, as you guys know, and we have talked about the flow from the heart to the ascending aorta, to the arching aorta, to the descending aorta, thoracic aorta, and then boom, abdominal aorta. Okay, so 20 to 25% of resting cardiac output. During exercise, this one drops down to 5 to 10%. And think about it, because we want that blood to go somewhere else where we're using it, such as the muscles. All right, so this one is just a frontal section illustrating the blood vessels of the kidney. Of course, if it's red, it's artery. If it's blue, it's vein. And we can trace one. So we have a renal artery. And then they're going to go into segmental arteries, all right? And then interlobar arteries, okay? And then they're going to arch and turn into the arcuate arteries. And you guys can see it here, okay? And then they're going to irradiate and turn into the cortical radiate arteries, also known as interlobar or lobular, I should say, lobular arteries, okay? So tracing it back, let's do it. Tracing back. We start with the renal artery, and there it's going to be segmental, and then it's going to be interlobar because it's in between the lobes. Then it's going to arch and turn into the arcuate artery, and then cortical radiate artery because they're going to be in the cortex and they're going to radiate. They are also known as interlobular arteries. This is where the filtration happens. They are going to turn into arterioles and then capillaries, and then from the capillaries, they are between the tubules. They're going to go back to the venules and then to the cortical radiate vein, then to the arcuate vein, and then to the interlobar vein, and then to the renal vein. Okay? Pretty important. Start to think about the places that we have talked about already: the medulla, the cortex, and of course this one is the capsule, okay? So this is where we're going to talk about the branching of the branching from the bigger to the smaller, right? So the renal artery is going to branch out into smaller arterial vessels called segmental lobar arteries, okay? And then they're going to become and branch out into the interlobar arteries because they're in between the lobes, right? And then they're going to branch out into smaller branches called arcuate arteries. Those arteries are called arcuate because they arch over the bases of the medullary pyramids, okay? So at this point we have talked up to this, guys. Now we're going to talk about the next ones, which are the smaller ones, and those will be the cortical radiate arteries, okay? So from the arcuate arteries, we're going to branch out once more into cortical radiate arteries. These arteries are also known as interlobular, not to be confused with the interlobar, okay? Interlobular, okay? Then they're going to branch out once more, and they're going to be from arteries to something smaller called arterioles, right? And they're going to go into the glomerulus. So the first piece is going to be called the afferent arteriole, okay? The afferent arteriole delivers blood into a structure called the glomerulus, okay? And this one is going to supply blood to very microscopic units called the nephrons. This is the basic structure of the kidneys, okay? So we have gotten to this point. We got all the way to here, and now we're going to go into the afferent or afferent arterioles, okay? Okay, so here's when things start to be a little bit more complicated, okay? So not all the blood that goes into the afferent arterioles are going to get into the glomerulus, okay? So some of that blood is just going to continue and go to other capillaries, and we're going to talk more about them, okay? So the vessels that got into the glomerulus have to come out, and they go out through the efferent arterioles, okay? So once it's into the efferent arterioles, blood is going to move into the peritubular capillaries. Think about it, peritubular capillaries. These capillaries are going to be around several pieces known as the convoluted tubules, either proximal, distal, or also the loop of Henle, okay? Then from there, the blood is going to go to the interlobal or cortical radiate veins, then the arcuate veins, then the interlobar veins, renal vein, and lastly the IVC. And then from the IVC, we know we go to the right chamber of the heart, also known as the right atrium. So now let's talk a little bit about the flow of the blood through the renal circulation, okay? So starting from the heart, we're going to go, of course, to the aorta. From the aorta, we're going to go to the renal artery, segmental artery, meaning each segment, interlobar artery, because it's in between the lobes, arcuate artery, and then we're going to get to the cortex, also the area where the cortical radiate arteries are. We already talked about the interlobular arteries because they are the same, right? And then we are going to branch out once more and turn into the afferent arterioles and get into the glomerulus, right? And those glomerulus or capillaries that they're going to go out as the efferent arteriole, okay? Those efferent arterioles are going to turn into either peritubular capillaries or vasa recta. They both have the same function. They're going to either reabsorb or secrete, okay? From there, we're going to go into the cortical radiate veins, arcuate veins, interlobal veins, renal vein, inferior vena cava, and then we're going to go back to the right atrium, right? To the heart, wash, and repeat, okay? Now we're going to talk about the anatomy of the kidneys and then the primary zones, right? So we're going to have a renal cortex, a renal medulla, and then where things start to drain down, the renal pelvis, okay? We already talked about the capsule, which is above. Then we have the renal cortex and then the renal medulla, and things are going to start to drain here from the renal pelvis to the ureters, right? So in the renal cortex is the outer zone of the kidney. This is the layer of the tissue that is light red and brown in color. The extensions of the renal cortex are also known as renal columns, and we'll see it in a little slide pretty soon, okay? And they project inward into the renal medulla layer. We see it here. This is the cortex, and then the renal column is this area here. They're just structural divisions, okay? Now, the renal medulla is the deeper layer that is dark reddish in color and is the zone where the renal columns are going to be, and also they're going to be separated by the renal pyramids, okay? We talked about the renal pyramids have a base, and so we're talking about the base, but also they have an apex. In this case, the apex is going to be called renal papilla, okay? And they're kind of like inverted pyramids. So we have it here, we have the base of the pyramid, and then the apex of the pyramid, right? And this is known as the renal papilla, okay? Base, renal papilla, okay? So then we have the renal pelvis. That is the funnel shape, and this is going to turn later into the ureter. So going inside the kidney, this renal pelvis is going to project in little cups, okay, known as calyces. We're going to have minor and major, okay? So we're going to have minor, then we're going to have major, and then we're going to have the pelvis, okay? And we're going to see it a little bit in the slide, okay? This is where urine starts to drain from the nephrons down to the ureters, okay? And we see it here, and this will make more sense. So we have the minor calyces here, then we have the major calyces, and then we have the renal pelvis. As you guys see, here is the nephron, where filtration starts to happen, and then through the collecting duct, we are starting to talk about urine that is getting drained here to the minor callus, then to the major callus, then to the renal pelvis, and then to the ureters, down to the bladder, and then out to the urethra, and then out to your toilet, right? Okay, so the renal calyces are this kind of cup-shaped structures. So there is a minor callus, okay, and this one receives the urine from the renal papillae. So we're talking about the tip of that pyramid, right? So then they drain to one major callus, okay? The major calyces are 2 or 3 tubular structures that are going to receive the urine from the minor calyces, and then these guys are going to drain into the renal pelvis, which is that funnel shape that receives everything from the major calyces, and then is going to drain finally to the ureter, right? And we see it here. So we start from the minor, major, pelvis, and then finally ureter, and then to the bladder, and from the bladder to the urethra, right? Depending if you're a man or a woman, there will be several structures that would be different. So now let's talk about the nephrons. So the nephrons are functional structural units of the kidney, because there is where the magic happens and we create urine, okay? So they help us filtrate, reabsorb, and secrete stuff that we don't need in our blood, okay? So the main parts of the nephron, we have the renal corpuscle, which is that area where we see all the convoluted capillaries and stuff where things are getting filtrated, okay? This one is located where? In the renal cortex, okay? And we're talking about glomerulus and glomerular, sorry about that, glomerular capsule, also known as the Bowman's capsule, okay? And then we have the renal tubules. Think about tubules, okay? They begin at the renal cortex, they also pass into the renal medulla, and then they return back to the cortex, okay? We have a PCT, okay? Proximal convoluted tubule. Then we have a nephron loop of Henle, or loop of Henle, and then we have a DCT, distal convoluted tubule, and then with a collecting duct where several nephrons are going to meet and drain the filtrate that is going to turn into urine. Here we see it, so we're talking about the renal corpuscle here, okay? So we have the glomerular capsule and the glomerulus, and you guys see it here. So the reddish part is the capillaries known as the glomerulus, okay? And there they're going to be encapsulated in the glomerular capsule. This is also known as the capsule of Bowman or the glomerular corpuscle, okay? This is what I think about as a juicer. Blood is coming in, things are getting squeezed, and again, things are getting filtrated. All this tubing system here are the tubules, okay? So we have, if it's closer, it's proximal, right? So we have first a proximal convoluted tubule, the PCT, proximal convoluted tubule. And then we're going to have a loop of Henle, also known as the loop of the nephron, okay? It has a descending part because, of course, it goes down, and then it has an ascending part, okay? We can talk also about a thick area and a thin area, okay? But for the purpose of this presentation, so long you guys know that there is a loop of Henle or a nephron loop that has a descending part and ascending part, I'm satisfied with that, okay? Then we have a DCT, okay? Distal convoluted tubule. At this end, we're gonna go to the collecting duct, okay? All the fluid contained in this area is gonna be known as filtrate, okay? Again, all the fluid that is being filtrated through the glomerular capsule in the glomerulus is going to be called filtrate, okay? Once we get here, we're going to talk about urine. So here what we're seeing are two nephrons, okay? The image can be overwhelming because we have a lot of parts put together here, but it's very straightforward, okay? First, if you guys see in the top, we have the names. We can all agree these two are nephrons, okay? It's just their anatomy, and they are just different because where they are located, okay? So cortical nephron, we can agree that this is closer to the cortex, okay? And then the juxtamedullary nephron is because it's closer to the medulla, which is here, but also its loop goes way lower than the cortical nephron, okay? They both have proximal convoluted tubules, okay? They both have distal convoluted tubules, and they all have loops of Henle. As you guys can see, the one on the right has a longer nephron loop or loop of Henle, okay? That's okay, but they both have the same similar functional structures, okay? Now, the purplish vessels that you guys see here, they're capillaries. Their function is to reabsorb, secrete stuff that we don't need. On the left, they're going to be called peritubular capillaries, okay? And on the right, they're going to be called vasa recta. They have the same functions. They are either reabsorbing or secreting, okay? They're both nephrons. They both filtrate. They both, at the end, are going to cook, figuratively speaking, and produce urine, right? Okay? Cortical nephron, juxtamedullary nephron. Just keep in mind, if it's juxtamedullary, medullary is going to have a longer loop of Henle, and its capillaries are going to be called vasa recta. If it's a cortical nephron, it's going to have a shorter descending loop or loop of Henle, and its capillaries are going to be called peritubular capillaries, okay? Now going to the renal corpuscle, we are going to talk about the capillaries that are inside that capsule. In that capsule, we're going to have the glomerulus. It's just, I think about like spaghetti or a piece of lasagna. It's just a ball of capillaries, okay? And this is where the blood is going to go and be filtrated, okay? These capillaries consist of fenestrated endothelium. They're also fenestrated capillaries, meaning that they have that cheese-like structure that's going to allow some stuff to come out and some stuff to come in, but it's just going to be a certain size. So we are still in the renal corpuscle. Now we're going to talk about the capsule, okay? So the glomerular capsule of the Bowman's capsule is the site where blood plasma enters the nephron, okay? Meaning what? This is water and fluid that enters into the glomerular capsule from the glomerulus, okay? So we're filtrating going to the space. This fluid is going to be called filtrate. It's not urine, it's filtrate, okay? So in the glomerular capsule is the location where we have for the first time filtrate. This is the raw material that we're going to use to make urine. So this is what we're going to use to cook urine, okay? The glomerular capsule has two layers. We have a parietal layer, and that's the outer layer, okay, that consists of simple squamous epithelium. Then we have a visceral layer. This one is an internal layer that contains modified epithelial cells called podocytes. These podocytes are called that way because they seem to have, or people think that they seem to have little feet, okay? So they're kind of like foot cells, okay? And they help with filtration. This improves and enhances how we filtrate stuff, okay? Then we have the renal tubules, okay? We have talked about this. This is where we leave the corpuscle, and now we're talking about the tubing, okay? Let's think about it that way, tubing, okay? So we first have a PCT, proximal convoluted tubule, and this is a dead giveaway because if it's proximal, it's closer to the corpuscle, right? Then we have the nephron loop of Henle, or loop of Henle, okay? With its descending and ascending piece, okay? And then we have a DCT, okay? distal convoluted tubule. This is the place that is further away from the glomeruli, right? Then after that, we're going to have the collecting ducts, okay? The collecting ducts is where several nephrons are going to drain into one collecting duct, okay? At this point, we're going to talk about urine, okay? So besides that, we know that there are capillaries surrounding these tubules, right? We have talked about the peritubular capillaries if it's in the cortical nephron, or vasa recta if it's in the juxtamedullary nephron, okay? These capillaries reclaim filtrate, meaning reabsorption, or they're going to get rid of substances that we don't need, known as secretion, and drain them back to the renal tubules, okay? And we see them here. Proximal convoluted tubule, PCT, loop of Henle, or nephron loop, and then distal convoluted tubule. We see them here again. So we have a PCT, PCT, DCT, DCT, and they all have a nephron loop. Also, those purple structures, let's focus the one on the right, known as vasa recta, okay? And it's doing the same thing. They're either reclaiming or secreting stuff that we need or we don't need into or out of the blood. They have a different name here. They're also known as peritubular capillaries, okay? Now let's talk about the peritubular capillaries. These are the capillaries that cling around the renal tubules, okay? This one is specific to the cortical nephrons, okay? They arise from the nephron arteriole, so this is like a continuation, and they absorb water and solutes from the renal tubules and put them back into the bloodstream, okay? We are reclaiming stuff that we need. On the juxtamedullary nephrons, they have a different name. They're called vasa recta, okay? Same thing, they only go deeper with that deeper loop of Henle, and they do the same thing. They reclaim water and solutes and put them back into the bloodstream, okay? We see them here. These are the guys, okay? Now we have the proximal convoluted tubules, okay? This is the section that is closest to the glomerular capsule, okay? This is also the longest and most coil of all four renal tubule sections, okay? Inside them, meaning in their inner layer, they're lined with cuboidal shape that epithelium that has microvilli. This increases the surface area, meaning that they're gonna be doing something akin to reabsorption, right? And this is important because if we improve surface space, we're gonna optimize area and reabsorption. Now the nephron loop, also known as loop of Henle, this is the U-shape area with a descending piece and an ascending piece, okay? And this is where we are. It has a descending limb, and this one is the one that is closest to the PCT. They also have an inner portion, of course, and it has cuboidal epithelial cells similar to the PCT, and the ending portion is simple squamous epithelial cells. Why is that important? Because probably in this area we don't have a lot of reabsorption, right? Then we have the ascending limb. That means that we're going up, right? This one is closest to who? The distal convoluted tubule. And then there's a change on the epithelium. We have cuboidal or columnar epithelial cells, so there is a degree of reabsorption or secretion. Then we have the distal convoluted tubule. So this one is the farthest away from the glomerulus, right? This one is attached to the end, as it says here, and also attaches to the collecting duct. It consists of cuboidal epithelial cells that have very few or almost no microvilli. Nevertheless, there's reabsorption here and also secretion, okay? Then the collecting duct is the area where we can talk about urine, okay? Several nephrons are connecting to this. So think about like a main sewer that is receiving waste from different nephrons, okay? There are two cells that are found in this area, okay? There are principal cells and intercalated cells, okay? So once in the collecting duct, we're talking about urine, okay? And they will exit the nephron through the papillae into the minor calyx, okay? These cells are pretty important because they are going to play a role in reabsorption or secretion of important stuff, okay? So the principal cells, they maintain water and sodium levels in the body, okay? They have sparse and short microvilli on them, okay? Then the intercalated cells maintain the acid balance of the blood, okay? So we're talking about pH, which is pretty important as we have talked about. They have cuboidal cells and many microvilli. So there is exchange of ions of hydrogen, also there's exchange of water, and there's also exchange of electrolytes. Here we see them again, okay? Cortical on the left, juxtamedullary on the right, and we're talking about nephrons. So now let's talk about the filtration through the nephrons. So starting from here, we're starting and filtering through the glomerulus, and now we're talking about filtrate. In the glomerular capsule, we're going to go to the PCT then, or proximal convoluted tubule, and then to the descendant limb of the nephron, also known as the loop of Henle. So at this point, we are at the U-shaped portion. So now we're going down, and then we're going up at this area, right? And then the DCT, distal convoluted tubule, and then to the collecting duct when we talked about urine. So urine formation, we talked about glomerular filtration, where we have filtrate. That filtrate is going to be reabsorbed, keeping a lot of the stuff that we need, and then we're going to have tubular secretion, meaning stuff that we don't need, such as drug metabolites or waste, okay? And water conservation, we need to keep a lot of the water that we filtrate. The amount of stuff that we are urinating is minimal compared to the stuff that we filtrate. So filtration versus reabsorption versus secretion. The filtration is the process of moving substance from the glomerulus into the capsular space, or also known as Bowman's capsule, okay? We're talking about filtrate at this point. So reabsorption is a process of moving substance from the renal tubules back to the bloodstream. So we are reclaiming potassium, sodium, other solutes that we need, and also water into the bloodstream via the peritubular capillaries or the vasa recta. Secretion is a process of moving substance from the bloodstream into the renal tubules, held by our friends again, the peritubular capillaries or the vasa recta. This is stuff that we do not need, such as drug metabolites or other kinds of waste. Okay, so now we're going to talk about the glomerular filtration. So it's plasma and solids moving from the glomerular capillaries into the glomerular capsule or capsule of Bowman, okay? So this is done by a passive process where forces are competing against each other, and it happens with the path of least resistance. The one that dominates on top of the other three is a hydrostatic pressure, okay? This forces fluid and solids to the filtration membrane, and then once they're in the capsule, this filtrate will travel into the renal tubules, okay? 99% of the material that is filtrated is what? Absorbed back, reabsorption into the bloodstream. This is how greedy we are, okay? So when there's trauma into the kidneys, meaning an infection or chronic trauma such as hypertension or uncontrolled diabetes that is gonna damage the capillaries, other stuff is gonna come out. Think about it. If you damage the strainer is not gonna be as effective, and if you damage it enough, bigger stuff is going to go through the strainer, right? So the composition of the glomerular filtrate has water, of course, because there's a lot of water. It also has some metabolite waste, urea, ammonia, uric acid, and creatinine, and some nutrients, okay? Glucose, vitamins, amino acids, and salts such as sodium or potassium. Red cells, white cells, platelets, and plasma should always remain in the blood because the fenestrations in those capillaries are too tiny to let these guys in the filtrate. If there's a damage, then is when we start to see these guys, okay? A lot of these guys might be reabsorbed, okay? Some will not, okay? So as we were talking, there are several forces that are going to help with the filtration. As we were talking, glomerular filtration is a passive process where forces act as the main driver of the filtration. The biggest of them all is going to be the hydrostatic pressure of the glomerulus, okay? This is the main force that is going to push fluid into the nephron, okay? There are going to be other forces trying to bring stuff back into the glomerulus. So we have the hydrostatic pressure of the capsular space attempting to bring back the fluid into the glomerulus, okay? And then we have the colloid osmotic, and whenever you guys see that, it's protein trying to bring same thing, fluid back into the glomerulus, okay? Of the three, the first one, the hydrostatic pressure of the glomerulus, is going to be the strongest, allowing the filtrate to pass through, and then we have filtrate, okay? All right, so overall, all these three pressures control the process of glomerular filtration, okay? All of them combined are going to be known as net filtration pressure, okay? So with the net filtration pressure, we have to think about just in simple terms, okay? That way it's easier to comprehend. So here I have a little bit of like doodling that I've done. So I have the outward pressures, meaning the hydrostatic pressure of the glomerulus. These are the same, meaning that the forces that are trying to bring stuff out of the glomerulus here, that way we can filter stuff here, but we have friends against it, or foes we should say, that are trying to not let the stuff to go through and bring it back, the inward pressures, okay? And these are the capsular space pressure and the glomerular osmotic pressure, trying to bring stuff back in the capillaries, okay? Of the three, the hydrostatic pressure is the one that is going to triumph all the time because it's stronger. Path of least resistance. If you guys make the math here, we can say that 30 plus 15 is 45, but this pressure over here, which is the hydrostatic pressure of the glomerulus, is stronger by 55, okay? Okay, so the glomerular filtration rate is something pretty important, okay? It's the amount that moves into the capsular space by both kidneys. The average GFR, or glomerular filtration rate, is 125 milliliters per minute. That totals 45 gallons a day, okay? Even this much amount goes through the nephron, a lot of this stuff comes back to the mainstream, okay? Why? To the bloodstream, I should say. The bloodstream, because we need a lot of this fluid, okay? And just a little bit of that goes out as urine, okay? So factors that control the GFR. Okay, factors that controls the GFR are simple. Changes in the filtrating pressures, meaning the hydrostatic pressure of the glomerulus versus the other two, right? Changes on the diameter of the afferent arterial and changes in the diameter of the efferent arterial, okay? Changes on the osmotic pressure of the plasma in the glomerulus, stuff that is going to try to keep stuff inside the glomerulus. Changes in the hydrostatic pressure of the glomerular capsule, meaning stuff that is trying to bring or trying to not allow that filtrate to go into the capsule. And also changes of the blood flow through the glomerulus, okay? Because if there are changes in that blood flow, some blood is not going to get filtered as fast. Okay, so there are different mechanisms, and we're going to try to talk about different mechanisms, okay? So this one is the direct regulation of the glomerular filtration rate by the muscle contraction of the arterioles, okay? So this one is known as the myogenic mechanism, okay? And it's managed by the smooth muscles in the wall of the arterioles of the blood vessels, okay? This one directly regulates the GFR, okay? By increased blood pressure, this causes the afferent arterioles to what? Constrict, meaning that those are gonna get smaller in diameter, okay? The vasoconstriction of the afferent arteriole, meaning the arterioles are gonna go in the glomerulus is going to help to maintain an ideal GFR. Okay? A decreased blood pressure causes the afferent arterioles to dilate, meaning the lumen is going to expand, allowing more blood to go in and helps to maintain an ideal GFR. The whole point of this is to keep a homeostasis, a balance, okay? Not too much, not too low. A balance, a sweet spot. And this is what we're talking about. This one affects directly the GFR to be increased when it's needed, either if the blood pressure goes down or if the blood pressure goes up, okay? Now, we're going to talk about the direct regulation of GFR by the tubuloglomerular feedback mechanism, okay? So the kidneys can also regulate the rate of, or GFR on their own, okay? There is a little collection of cells, they are called juxtaglomerular apparatus, okay? They are formed from the nephron loop against the afferent arteriole that supplies the glomerulus, okay? These group of cells have different names. There's one that is called the macula densa, okay? In this area controls the filtering function of the glomerulus by changing the diameter of the afferent arteriole. So we are still doing some constriction here, okay? Also, these cells are pretty important with the production of a hormone called renin, and we're going to talk about this guy later, okay? So the direct regulation of the GFR by the tubuloglomerular feedback mechanism is basically the following. The JG apparatus can affect directly into the afferent arterioles by constricting and then either increasing or decreasing or increasing the GFR, okay? So, a little bit more here, the macula densa can cause what? Vasoconstriction or vasodilation. So, given the necessary, I can either decrease the GFR and the blood flow or increase the GFR and the blood flow, okay? Kind of like a myogenic function, but this is done directly by these little cells here, okay? And we see it here. And also this one directly affects the GFR, okay? Now let's talk about the indirect regulation of the GFR done by the nervous system. In this case, it's the sympathetic nervous system. When we think about this one, usually it's about the fight or flight response, okay? So the sympathetic is a division of the autonomic nervous system that triggers the smooth muscle of the arteries to contract, which directly increases what? Blood pressure, okay? Because the blood pressure is increased, the GFR also increases, okay? So therefore, GFR is indirectly increased by the increase of blood pressure with this mechanism. We are not talking that's something that is directly related to constriction of blood vessels per se, or by the GJ apparatus. This is by the nervous system, okay? And we see it here. Okay, this one increases not the GFR alone, but the systemic blood pressure. Okay, now this one is the fun part. We are going to talk about the indirect regulation of the GFR by our good friend the renin, angiotensin, aldosterone system, also known as the ROS pathway. So starting from the beginning, renin is a hormone that we talked about that is produced directly, specifically in the GJ apparatus, right? For this purpose, we're going to say kidneys, okay? So when times of low blood pressure or low blood volume, the renin is going to be produced in the kidneys and is going to interact with a hormone in the liver called angiotensin 1, okay? Angiotensin 1 is an inactive form of a hormone, okay? This hormone has to go through the bloodstream to the lungs and interact with an enzyme called ACE, which stands for angiotensin converting enzyme, okay? Once this hormone interacts with ACE, it's turning to angiotensin 2, okay? Still. And then this one is an active form, but we're not done yet, okay? The process of angiotensin 2 in the bloodstream is going to cause the adrenal glands to release a hormone called aldosterone. Okay, the aldosterone basically is going to go back to the kidneys and more specifically to the DCT, okay, in the collecting ducts and increase water and sodium reabsorption and increase potassium secretion. So we are exchanging potassium for sodium and water. The end result of this is that we're going to increase our volume and our pressure and keeping a good balance. So we increase our blood pressure, increase our volume. That way we are achieving a balance in our blood pressure. Okay? So now we have the renin, angiotensin, aldosterone pathway a little bit more like in a graphic sense, okay? So we have a trigger, we have a reason why our volume is going down. We either dehydrated, our sodium is low, or we're bleeding out, okay? So all these guys are going to result into what? Our blood pressure to go down. The decrease in blood pressure is going to stimulate the JG apparatus to produce renin, right? Then they're going to interact with angiotensin and they're going to produce angiotensin 1. That angiotensin 1 is going to go to the lungs and interact with the ACE, also known as aldosterone converting enzyme, and produce angiotensin 2. Okay, this angiotensin 2 is going to go to the adrenal cortex and is going to produce and increase the production of aldosterone, which goes to the kidneys in the distal convoluted tubule and the collecting tubules or ducts, I should say, collecting ducts, and increase the absorption of water and sodium, right? Then that increases our volume and then blood pressure is achieved until everything returns to normal and we achieve balance, okay? So now we're going to talk about tubular reabsorption. This is a process where the substance is selectively moved from the renal tubules to the bloodstream, okay? Through our friends, either the pituitary capillaries or the vasa recta. Most of this reabsorption happens in the proximal convoluted tubule, okay? Anything that is not reabsorbed will become at some point urine, okay? Mechanisms of reabsorption: water through osmosis, and we talked about this in class. Sodium through active transport, meaning these processes need ATP, right? All these guys. Same with glucose, same with amino acids, and same with vitamins. Proteins basically is with pinocytosis. Basically, they're getting a little bit engulfed and transported through the cell and then back to the bloodstream, okay? So reabsorption of materials is limited. There's only so much that can be reabsorbed. So there is a point when we reach a maximum and then we plateau, okay? So hormones that control reabsorption of water and other solutes are pretty important, okay? So there are two hormone pathways that help regulate the movement of fluid in and out of the renal tubules, okay? Those hormones are created in other places of our body and they act upon some target cells, okay? So the first one is the antidiuretic hormone, also known as ADH. This one is released, I repeat that, released by the posterior part of our pituitary, okay? And then we have talked about the renin, angiotensin, aldosterone pathway produced in our kidneys, more specifically the JG apparatus, okay? Now let's talk about the hormone control reabsorption done by the antidiuretic hormone, okay? This one is released by the pituitary gland when we have decreased blood pressure, increased solute levels, meaning there's a high concentration of solids in our blood by the cause of low fluids, and then decreased blood volume. The whole stimuli of this is like our blood pressure is, or volume level is low, right? So this hormone travels through the bloodstream and it targets the principal cells where in the collecting duct. They become more permeable to water, so things get reabsorbed, and water is retained in the body. The end result is the urine output is reduced. And if there is a way to memorize this hormone, is anti is against diuretic, which diuretic means diuresis equals urine. So it's the hormone against urinating. Antidiuretic hormone. Okay? Then we have the reabsorption of fluid through the renin-angiotensin-aldosterone pathway. So renin is produced in the kidneys, then it's interacting with angiotensin 1 in the liver. That one releases angiotensin 1, and it interacts with the ACE or angiotensin converted enzyme in the lungs, turning into angiotensin 2. Then angiotensin 2 goes to the adrenal glands and it turns into aldosterone. Aldosterone, again, is going to interact with the distal convoluted tubules and the collecting ducts to increase water reabsorption and sodium reabsorption, okay? And increase volume pressure. We've seen this guy before. I'm not going to talk about it again. Tubular secretion. So we are not reabsorbing. We are getting rid of stuff, okay? This is the process where substances are selectively moved from the peritubular capillaries, meaning the blood, to the renal tubules, okay? So we are getting rid of stuff that we don't need. Usually these are chemicals, waste, drugs, and unwanted materials that we don't need in our body and we need to get rid of it, okay? This happens at all parts of the renal tubules and the collecting ducts, okay? The water needs to be conserved, okay? Water is conserved through all these mechanisms that we have talked about, such as antidiuretic hormone and aldosterone, okay? The end result of this processes are going to create urine, okay? A little bit more exemplified here. So we have the renal corpuscle here and then the tubules, right? So the efferent arteriole is going to come in and then we're going to start to filtrate here, right? We're going to create filtrate. Those capillaries in the glomerulus are going to turn at some point into the efferent arteriole, and we're going to continue our merry way to either be peritubular capillaries or vasa recta, right? So we're going to reabsorb fluid from the tubules to the bloodstream, or we're going to get rid of stuff, right? Through secretion. And at the end, we're going to create urine. Okay? So urine, when it's secreted from the body, it usually is yellow to dark yellow and transparent. You can see through urine, right? Slightly aromatic, and it should not smell heavily because that should be an indication of something else. It's slightly acidic, but the pH ranges between 4.5, which is very acidic, to maybe alkaline, okay? The specific gravity is from 1.001 to 1.035, and this is just a measure of ratio of mass of specific substances compared to the mass of distilled water, okay? This basically indicates that urine is slightly heavier than water, okay? The more hydrated you are and the more water you drink, the specificity of urine is going to decrease, okay? And there's a lot of stuff that's going to influence this, meaning, for example, if you're pregnant, or if you have uncontrolled diabetes, or have liver disease, or use drugs, or you have a terrible infection, right? So urine components. So components of urine, 95% of the urine, the volume, is water, okay? And then we have urea, and then we have uric acid, creatinine, and other electrolytes, sodium, potassium, phosphate, and sulfate. Things that should not be in urine. Glucose, proteins, red cells, and white cells, because that means something is off, all right? Maybe it's an indication of uncontrolled diabetes. Maybe it's an indication of a heavy injury to the glomerular capillaries and nephrons, and then we're letting stuff filtrate through. Infection or damage, these are bad things that we don't want to see and need to be addressed. This is kind of like the first signs that something is amiss. All right, so kidney volumes. So a typical adult processes around 180 liters of fluid per day, but look how much we only pee out, 1 to 2 liters, okay? So now let's talk about the odds. Polyuria is when you urinate more than 2 liters in a day. We pee a lot, in a pathological way, let's say. It's not normal. If you're drinking a lot of water, well, that can affect, but if you're not drinking a lot of water and then you're still urinating a lot, something is amiss. Oliguria is less than 500 ml of urine in a day, scantily of minimal urine. So we either dehydrated, we have an injury in the kidneys, or something's going on that is not letting us urinate, right? And anuria is when less than 100 mLs of urine happen per day, or no urine. So basically, you're just not urinating, you're retaining fluids, and we have to address the problem that's going on. Factors that affect the urine output, as we were talking before, a lot of fluid. Of course, if you drink a lot of water, you're gonna pee more. Temperature, of course, humidity. If we are too excited, we see it with our little dogs when they get too excited, they pee, right? Respiration rate, of course, and body temperature. Either we're cold, too hot, we're gonna pee. All right, so now looking at this, we're gonna talk about different diseases that can affect what's present in our urine. We talked about this in class. I'm not gonna go over this, okay? So now urine. Urine is gonna be passing through the collecting duct, going through the papilla and then to the minor calyx, major calyx, renal pelvises, ureters, urinary bladder, urethra, the external urethral orifice, and then the outside, meaning your toilet or a tree or whatever you are going to be on. All right, the ureters are slender muscular tubes that carry urine from the kidneys to the bladder, okay? They have an inner mucosa, a muscular layer, and a fibrous layer. Sounds familiar? We've seen it many times with other stuff, okay? Mucosal layer consists of what? Transitional epithelium, and you guys will see this very specifically always in the urinary tract, okay? The muscular waves of the urethra and the gravity is what is going to move the urine into the bladder, okay? And then talking about a little bit more about from the ureters to the bladder. So the bladder is the sac that is located in the pelvis, okay? And it has a mucosal layer that consists of transitional epithelium, see? And then it has smooth muscle called the trousseau muscle, okay? And then it has a fibrous adventitial layer, okay? The urinary bladder has also, when it's not full of urine, has a rugosity epithelium, also called rugae, okay? When it fills up, of course, that's gonna be smooth, right? Okay, there's also a piece called the trigone, okay, that is formed by the bladder neck and the orifices. This is called the trigone, okay? This is the place where a lot of infections occur, okay? Now we see the difference, female structures on the right, male structures on the left, okay? As we can see, we have the similar structures to some degree. We have a bladder in both cases. We have rugae. We have an inner layer, a middle layer, and external layer. We all have urethral orifices, and we both have either internal sphincters and external sphincters, meaning involuntary, involuntary. What changes in the male is that we're going to have a prostate and we're going to have spongy body. So our urethra is either going to be called urethral, prostatic urethra in the prostate, and then membranous urethra by the membranous of the pelvic floor, and then spongy urethra when we are around the spongy bodies, right? Okay, so the urethra is the terminal structure. So we have an internal sphincter, which is smooth muscle, meaning involuntary, and external urethral sphincter that is voluntary, okay? And then at the end, we're going to have a orifice, an external urethral orifices, okay? So structural differences between the male and the female. We talked, female is always shorter, male is always longer, and the male has a prostatic urethra, membranous urethra, and a penile or spongy urethra. This is one of the reasons that females are more prone to UTIs, because they have a shorter urethra, and also the proximity to the anus makes more ideal conditions to get a UTI. Males can get UTIs and they get it more often, but in cases of incidence, females get more UTIs than males, given this. This is one of the reasons that it's recommended after sex to urinate and make sure that you don't get a UTI, okay? Here you go. Structural differences. So now the micturition reflex is the process discharging urine from the bladder, okay? When the bladder is distended around, and I should not say, 200 mLs it's like the minimum that stretches the receptors and creates that reflex to tell your body we need to pee. Okay, so the micturition process. So there's the stimulation, there's contraction of the detrusor muscle of the bladder, and then the internal urethral sphincter, which is the internal one, is going to force open. It's going to relaxate the external one until you cannot hold it anymore, and then it goes out. Here we have the diagram. You can take a look at it. We have talked about it, and this is pretty much it about our urinary system. I appreciate your time. We'll see you tomorrow. Thank you. Bye-bye.